So hello everybody. So my name is Kevin Singh. I'm a graduate student from Dr. Andrew Su and Dr. Chun Liu's lab. And today I'm mainly going to talk about BioThings Core as well as Smart API. And the main topic is about navigating connected API networks. So the main topic today is about APIs. So APIs actually stands for Application Programming Interface. It has been very widely used in the whole society in multiple different scenarios. For example, like API has been used to power like web applications, to power like mobile devices. So the major advantage of APIs is it provides a very efficient way for people to distribute their data. So despite its application in all kinds of different domains, it also has gained major interest in the biological community. So previously, like maybe like 10 years ago, when people have their database released, they normally have a data dump, so people could download a copy of themselves either through like a CSV file or like a SQL database. But right now, we have seen like increasing efforts from a lot of biological teams to provide APIs as their data access points. So some of the examples may include some major data providers like Uniprot, NCBI, or like BI. And there are also efforts from data aggregators who like aggregate data from different databases. So such efforts include like BioThings APIs, which aggregate information from like gene annotation or variant annotation data. So they all try to distribute their resources through APIs, which returns JSON output formats. So here's just some example of the current available APIs in the whole biological community. So for example, like MyGene.info or like DGIDB, which stands for like Disease Gene Interaction Database. So, but they only represent a very tiny fraction of the large amounts of APIs currently available. So it's good to have so many APIs available to use, but the problem is that we don't have a central registry system where we can find and locate all those APIs. So for example, let's say like I want to get information about the gene-disease relationships. I don't know like which APIs I actually can use. And the second problem is that even you know that which API I want to use, sometimes you may have the issue. You might run into the issue that some of the APIs are not very well documented. So you don't know even you don't even know how to make API calls. So these are two major issues for the current development of APIs in the biological community. And besides, once you make the API calls, you're going to get some outputs, either like XML or like JSON. I'm mainly talking about like JSON out formats here. So one key thing about JSON is that JSON really lacks schema. That is to say that JSON file itself doesn't provide any information, any like a semantic information about what each field is about. So here's an example of like 3 different API outputs. So the first one is from Example, right? So you can see that these 3 API outputs actually all contain information about the same thing, which is an Example gene ID, which is highlighted in the yellow one here. But you can see that all these different APIs, they try to represent their data in different names. For example, Example use ID, while like MyGene.info use gene, and MyVariant use gene ID. And you can say that in MyGene and MyVariant, Example gene ID is actually nested under another different field. So this makes a problem because it's hard for us to know that which field what's the representation of each field, and it may occur problems that we could not systematically automatically extract the output which we want. So these are the current major problems of the development of APIs, and that's why we started the Smart API project. So Smart API project itself first is a specification. So it's an extension on the Open API specification, which provides like documentation of the metadata information of APIs. So in case you're not familiar with like Open API specifications, on the left, this is an example of Open API specification for MyGene.info. So you can see it provides lots of like metadata related information. For example, like server information, what the server of the API is, as well as information about the API endpoints, and also like information about the parameters of the APIs. For example, this API MyGene.info use gene ID as one of its parameters. And by taking advantage of all those information like server endpoint parameters, as well as like HTTP method, for example, here it's the HTTP method is shown to be get. So with all those information, you can actually construct, very easily construct your own API calls. For example, here, I know that this API takes Jing ID as its input. So I can just replace Jing ID with NCBI Jing ID 1017. And you can actually retrieve a JSON document directly from my Jing.info. So you can see that here is once you import 1017, you use OpenAPI to construct the API calls and you can get the API results. So that's the good thing about OpenAPI. So it basically provides very basic metadata information about the API. So you know how to construct API calls. And Smart API, it actually extends what's currently contained in OpenAPI specifications. And the cool thing about Smart API is that it provides semantic information inside of the specification. So for example, if we look at the example earlier, it only says that this ID is a Jing ID, right? But we don't know what exactly Jing ID it is. It could be an NCBI Jing ID. It could be a Jing ID from Ensemble. So the Smart APIs, what it does is it provides semantic information. So for example, here, you can see it provides some extra fields like X value type and X response value type. So it basically tells you that what is the input of the API and what is the output of the API. And the values of these value types are actually URIs. So URI actually stands for Universal Resource Identifier. So they're unique for all concepts. So each concept has its unique URI. For example, like NCBI Jing ID has a unique identifier from identifiers.org as well. So based on this information, we can easily understand that this API actually takes an input either from NCBI Jing or from like Ensemble, and what it outputs is like Uniprot IDs or like React Tom IDs or like Wiki Pathway IDs. So Smart API, like by extending those, by adding those semantic information, we can easily know that more like input-output information about the APIs. So that basically, with Smart API and Open API, we basically solve the issue of like making API calls, API input and output, but we still don't solve the issue of API interoperability, right? That is like different APIs have different outputs. How do we like semantically align those outputs together? So that's where like JSONLD comes into place. So JSONLD actually stands for JSON for linked data. And what it does is that because we know that we have very little control of what API output is, because it's wholly controlled by the data provider to do whatever they want. But JSONLD actually allows you to have a flexible way to modify the API outputs on the fly. So what it actually adds is you add a context file for API. So you will create a JSONLD context file for each API endpoints. And what the context file records is the field semantics of the fields. For example, Ensemble, what Ensemble is about. So Ensemble is identifier.org example. So you provide a URI for each of your fields. And once you provide this information, and JSONLD has its internal algorithm, which can automatically convert it from a normal JSON document to a JSONLD document. So you can see that right now, these Ensemble IDs, Ensemble and symbol, all those field names are actually replaced by URIs. So let's say you have another API which outputs like Ensemble ID, right? So all you need to do is change your context into Ensemble ID, and you will all get the same results. So it basically semantically align. It provides a very easy way to semantically align all different API outputs. And this is only a very simple version of the JSONLD context. You can also like embed a lot of like relationship information. For example, what's the relationship between a gene and a drug? You can all embed those relationship information inside a JSONLD context to make it more rich. So that's so far for JSONLD. And what Smart API also offers is a central registry for APIs. So for example, so you can like have your own Open API specification stored on your own server, but once you put your URI for your Open API Smart API specification here, you can register it in the Smart API. And once you register in the Smart API, your API could be searchable from the Smart API interface. So people could know that, okay, how many APIs we currently have, what each API is about. So they provide a central registry system for people to locate, to find API of their own use. So that's the summary about Smart API. So first, it provides a very standard way for people to document their APIs. So people can easily know how to make API calls. People know what input and output of each API is about. So the second part for Smart API is it provides a great way through JSONLD to semantically align different API outputs. So APIs from API outputs from my gene.info, my variant.info, or about example gene ID could be semantically aligned together. And the third, it provides a central registry for all biological APIs. So once people register here, we provide a central way for people to search all APIs. So currently we have over 25 different APIs registered in the Smart API. And if you have your own APIs, please feel free to register in the following link we provided. And so Smart API basically, so that's so far for Smart API. And but that's not really about knowledge itself, because we provide metadata information. But what can we do with this metadata? So, for example, especially in the case for BD2K, we want data to lead to real knowledge. But that is to say, can we actually link those data from different APIs together? Because previously there has been a lot of efforts, for example, data mining efforts, who try to do data exploration from all kinds of different domains, gene information, variant information, disease information. You group everything together and do data mining. And most of the efforts has been what they're trying to do is build a local database, which parse all the databases together. But the actual invention of this API actually provides us a unique way, so that we no longer need to do our own data parsing, not to say maintaining all those data up to date, but we still can take advantage of all the data provided by those data providers. And the way is through linking all those APIs together. So you can see that, for example, in the case here, my gene.info, right? It has an input. One of its inputs we know from the OpenAPI specification is that it has NCBI gene ID as an input. And we also know that once it takes NCBI gene ID, two of the possible outputs is HGNC ID or HGNC symbol. And we also know there is another API, which is disease gene interaction database. And what it takes is HGNC symbol. So once it takes HGNC symbol, what it outputs is a Campbell ID. So it's basically convert from so it basically returns all the Campbell ID, which represents drugs. So drugs related to these genes. So we can see that the input of DGIDB is actually the output for my gene.info. So you basically can link those API together through HGNC symbol. So you can connect from any NCBI gene ID directly to Campbell ID through HGNC symbol, right? So similar thing is that, for example, on the bottom, you can see this BioLink API. One of its input is HGNC ID. And one of its output is disease ontology ID, right? So it connects you from a gene to a disease, the disease-gene relationship. But my gene.info doesn't contain any information about disease. But what you can do is actually, because the input of BioLink is actually the output of my gene, so you can actually connect my gene and BioLink together through HGNC ID. So ultimately, what you get is you can directly go from a NCBI gene ID to a disease ontology ID. So you link from NCBI gene to UID. And if you group all those nodes together, you can actually create a connected network of different APIs. You can connect all those biological APIs together. And the ultimate goal is that you can actually connect the data behind these APIs together. So you can actually connect information about the gene, information about chemical, information about drug pathway all together. So you create a network of information data from different domain knowledges. And that's what the BioThings Explorer project is about. So BioThings Explorer basically takes the information provided by Smart API about each individual API endpoints, and they try to interlink those APIs together for knowledge exploration, so people can connect from like genes to drugs to diseases. So they provide a central way for people to interlink all those data. And then I will give a short presentation live demo of what BioThings Explorer is about. So, so as you can see here, this is an interface for the BioThings Explorer. So you can see on the first graph, like on the landing page, what you can see is a graph directly built on top of all the open the Smart API specification as well as the JSONLD context involved. So you can see that different color represents different semantic type, like genes, proteins, variants. So to do this graph, we basically group all those like identifiers from the input and output of the API together, and also like provided from the JSONLD, we extract the relationship information about the input and output, and we are able to create a central graph to show that how different biological knowledge are connected together. A similar view we could do is like through like a biological ID view. So for example, this is basically expand on the semantic type, and what you get is each individual biological IDs. So you can see that. So right now, like the different colors represents different semantic types. So for example, the pink ones represents like disease IDs, and the blue ones represents gene IDs, while the green ones represents like pathway IDs, and the red ones on the bottom actually represents all like different drug IDs or chemical IDs. So you can see a central graph, a central map showing you that how different biological entity IDs could be connected. So if we say like I want to go from let's say like ARPANET to HGVS, you know that okay, you can go through my variant and you can get the information. So once you hover on the edge, you actually can see like what's the API endpoint which can do the job. And if you enlarge it, you can see like what's the actual relationship between different biological IDs. For example, like HGVS actually is associated, ARPANET is associated with the variant HGVS. The similar thing for like HGN symbol, you can see a lot of different relationship going on here. And also there's another view which is like on the API level. So here we basically cluster all APIs together based on their inputs and outputs. So different color represents, so the blue represents APIs, and different color represents different API inputs and outputs. So you can see, for example, like if we enlarge this, you can see that my variant actually like connects a lot of like variant related IDs like HGVS or only variant ID, as well as DB-SNP, and like it can actually be connected from my variant directly to a Monarch API through HGNC, because HGNC is actually the input of this Monarch API, and it also happens to be the output of my variant. So you can connect data from my variant directly to like an API from Monarch. So that's a general overview of like how different APIs could be connected together. So once you have your own like a Smart API specification ready, you could register it in Smart API, and we could automatically help you to populate your own API onto this graph to provide a more detailed network of different biological knowledges. And also what it also provides is it also provides you with a visual view of API metadata. For example, like if you're interested in an API like MyGene.info and you don't know what it is about, you can click on any of this. Yeah, and you can see it will automatically create a graph telling you that what the API and input is, what's the output of the API is. So for example, on the left side, you can see these are all represents API inputs. So you can see that myCan, this is a mycan.info API, and it's the input of the API is like a drug bank, like UniProt IDs, like SNOMED IDs, like Uni IDs. And what the output is an inch key. So you can convert from like drug bank to inch key. And like different colors actually represent different semantic type. You can like look at color schemas here. So besides on the endpoints, like there's also one more use cases like sometimes people are interested to say like what if I have a UniProt, like which APIs can take a UniProt ID, or like I have an NCBI gene ID, like which API can take an NCBI gene ID. So for example, if you search for like a UniProt ID, let's say like UniProt here, it will automatically shows you like how many APIs actually take UniProt. For example, here you can see that there's an API called mygene.info which takes UniProt, and the outputs like HGNC symbol and CPI gene ID. And there's also another API like mycam, so it can take UniProt and outputs InchiQ. So also like other APIs like myvariant, which can take UniProt and outputs HGBS IDs. So this is one way for you to see like what you can do with your own IDs. And another way you can see it is that if I want to get something like NCBI gene ID, what do I need to give the API? So you can see that this will show you like all the different APIs which can connect to NCBI gene ID. So for example, you can see here on the right side, it represents the output. So the output we set to be NCBI gene ID. It's going to tell you there are 5 different APIs which can connect to NCBI gene ID and what each API actually takes. So yeah, this is a very like an overview of like how you can do with all this metadata. So besides like providing metadata information, as I pointed earlier, what we are ultimately want to achieve is like for data exploration. So how can we do data exploration? So from the example I gave earlier, so maybe let's say like we want to go from NCBI gene ID, and so I have an NCBI gene ID, but I want to get all the information about the drugs related to this gene ID. So you can say like a Campbell drug. So this is basically you tell BioThings Explorer that I want to start from NCBI gene and I want to get Campbell ID. And you can also like specify like how many APIs you want to connect, because you can connect like multiple different APIs together to finally achieve your goal. So for example, if we set the maximum number of APIs to 3, it will automatically help you create this kind of like a Sankey plot, which shows you like how different APIs can be used to connect different biological IDs. For example, like you can see that NCBI gene ID is on the most left side is your input, and on the most right side is your output. So you can see it's a Campbell ID. So all the red things represents APIs. So you can see that there's one API like mygene.info, right? It can connect you to like HGNC symbol. Then from HGNC symbol, it's going to connect you to DGIDB, and from DGIDB, you're going to get the Campbell ID. So that's basically connecting from a gene ID to a gene symbol, and then finally from a gene symbol to a Campbell ID. There are all kinds of like different passes. It will automatically populate with you all kinds of different possible passes which connect from different biological IDs. So you can also like I said, like you can specify, maybe you say that this graph there are too many different passes. You can choose like number 2. So that's basically say at maximum, I will use 2 APIs. So you can see this will bring you a like more clean graph. So now you can see, but the data is actually still the same. You can see NCBI gene can be first connected to HGN symbol. Then you go to DGIDB and you get your final results, which is a Campbell ID. And also you can view it in like a hierarchical view. So for example, you can see here, it's going to tell you that all those 3 different passes, which connects from NCBI gene to Campbell. So the first pass is from NCBI gene through a database called human base, and you get HGNC symbol, and then finally it gets Campbell from DGIDB. But you can also go through like mygene.info, get symbol, and then get Campbell. Or you can even more straightforward, you can go directly from NCBI gene through DGIDB and get Campbell. And one more thing about the BioThings Explorer is that it's not only allow you to like visualize how APIs connected, you can actually do the API calls, because OpenAPI gives you the capacity to like perform API calls automatically. So let's say that I want to choose like pass 2. So you can select pass 2 here, and you can provide any of your like inputs which you like. So basically any NCBI gene ID, you can put it here. It will automatically make the call to mygene.info and get an HGNC symbol, and then get Campbell ID through DGID. So for example, we put 1019, NCBI ID 1019 here, which is NCBI ID for, I think, CDK4. So, and then you can click on like explore this path. So it will automatically run API calls for you. So you can see what it actually does is it actually connects you from 1019, which is your input. It guides you to HGNC symbol CDK4. That process is done through mygene.info. So you can see the relationship information is also on site. So basically, NCBI gene has an XREF called HGNC symbol CDK4. And from CDK4, that's done through DGIDB. So you can see associated with chemicals. So CDK4 from DGIDB, you can connect to all those different chemical IDs through DGIDB. So BioThings Explorer, it basically takes all the information from Smart API specification as well as the JSONLD context file, and it helps you create a very equal network of different biological data, different APIs. So that's so far for the demo for BioThings Explorer. So yeah, if we also have a tutorial, so you can click on it and like explore all those different functions in BioThings Explorer. So yeah, I think that's so far for my presentation. So last, I would like to thank my advisor from Dr. Cheng Lei and Dr. Andrew. Also our team members in TSRI. This is also a collaboration between TSRI and the Maastricht University, which is led by Dr. Michelle. And I would also like to thank other staffs from API Interoperability Working Group, as well as funding support from BD2K. Yeah, so that's so far for my presentation. If you have any questions, please feel free to ask.